We have been lucky enough to participate to the development of so-called CAR T-cell, chimeric antigen receptor T-cells in bilineage ALL in children and adolescents. The development of this drug have been based on a multi-center intercontinental trial called ELIANA. And after this trial, who led to the approval of the product in 2017 in USA and 2018 in EU, there was a kind of expanded access program, but within a trial called B2001X trial from the company named Novartis, who is developing the Tisagenlecleucel in pediatric ALL, and also they develop it in adult lymphoma, which is not the topic now. This trial, 2001X, have authorized some criteria which were not possible as inclusion criteria in the previous trial, ELIANA. And that's why we were interested to look at the data with the other investigators of the trial. We have, if I remember well, 9 sites. And these 9 sites have looked together at the data in terms of what happened to children who were pre-exposed to another drug targeting CD19, which is the Blinatumomab. And secondly, what happens to the children who were exposed between the apheresis and the infusion of CAR T-cell to a drug named Inotuzumab, which is an antibody anti-CD22 antigen, coupled or conjugated with a chemotherapy named calicamycin. Inotuzumab is a drug which is very interesting in ALL. The problem potentially is that as it's very effective and brings some patients to minimal residual disease, we could be undetectable in some patients. Also, it destroyed normal B cells. So the question we were asking were the following. One, for Blina, does the Blina increases the probability of clonal selection towards the selection of clones without the target CD19, so-called CD19 negative cells? And question 2, do the patients who receive Inotuzumab have had less expansion and less persistence of CAR T-cell, and finally response which were inadequate? Unfortunately, there are not enough patients to answer clearly to those patients, but the abstract you can see in the EHA website, which will be, and the registered session will be available on Friday, next Friday. This abstract will show you that there is some trend for thinking that the overall results, if patients are pre-exposed to Blina or if patients are pre-exposed to Inotuzumab, are maybe inferior to the one of the patients who did not receive any pre-exposure to this drug. So I repeat, Blinatumomab, somewhere in the history prior to exposure to CAR T-cell, and Inotuzumab close to the lymphodepletion, which is the part we call bridge to CAR between apheresis and infusion of CAR T-cell after lymphodepletion. This was the main target of the study, but the other targets were, do a program with expanded access can reproduce the excellent result obtained in the ELIANA trial in terms of event-free survival? And will the toxicity data generated in ELIANA will be reproduced also? And you may know that these CAR T cells are very effective, but also very toxic because of the induction of so-called cytokine release syndrome and neurotoxicity. What we observed was that event-free survival at 12 months was even better in 2001X compared to ELIANA. We didn't make front-to-front comparison, but the data are at least as good or maybe superior to the one of ELIANA trial. And second, the toxicity was also inferior in terms of grade 3/4 CRS or grade 3/4 neurotoxicity. We think that this could be due to at least two factors. The first one is that now the teams which are dealing with these techniques and children are much more experienced, and they know how to bridge to CAR and probably which lead to bring to CAR patients in a better tumoral status. This is one point. And the less tumor you have, the better the tolerance. And second, the way to manage toxic effect also has progressed. And for example, obviously the two factors I'm referring to are linked, but for example, the ICU transfer was reduced compared to ELIANA. So we have made progress in terms of how to bring these patients to CAR T-cell. And this has resulted in probably better response a little, but also, and maybe more visibly, better managing of side effects.